Hi. Mendel didn't know what the molecular basis of inheritance was. It was not until the 20th century that the discovery of DNA solved this mystery, opening new pathways for discovery in biology. Today we are going to discuss DNA's discovery and learn what DNA is. And now, here comes the voiceover. Today we're going to talk about the story of DNA. And this image, photo 51, by Rosalind Franklin, played a critical role. And this is an image of the X-ray diffraction caused by DNA, and we will discuss this as the lecture goes on. In the 19th century, many people were eager to find what the genetic material, what the basis of heredity was. A genetic material needs to have certain characteristics. Genetic material must contain complex information. It must be capable of storing large amounts of information that includes the instructions for the traits and functions of an organism. Genetic material must replicate faithfully. Genetic material must have the capacity to be copied accurately. Every organism begins life as a single cell. To produce a complex multicellular organism, this single cell undergoes billions of cell divisions, each of which opens the possibility for mistakes in the copying of genetic information. Genetic material must encode the phenotype. The genetic material must have the capacity to be expressed, to code for traits. The product of a gene is often a protein or an RNA molecule. So there must be a mechanism for genetic instructions in DNA to be copied into RNAs and proteins. And this would be discovered in the 20th century. Genetic material must have the capacity to vary. Different species and even individual members of a species differ in their genetic makeup. This is essential for evolution, this capacity to vary. So during the 19th century, a good deal of progress was made. DNA was discovered in the nucleus. Mendel did his classic work on the segregation of genes. We had the development of the cell theory. We began to recognize that the nucleus was the site where heredity was contained. As the century progressed, we started to learn more about what DNA was. So DNA was discovered in 1868 by Johann Friedrich Miescher. He wanted to be a priest, but he became a doctor instead. He got interested in the chemistry of pus. He extracted the substance in the nuclei of pus cells obtained from discarded surgical bandages. He called the substance nuclein. He noted that the presence of a material that had a high percentage of phosphorus was in this nuclein, and he separated the substance into a basic part and an acidic part. This substance was later named nucleic acid by his student. Other discoveries of note in the 19th century, as I discussed, was that the physical basis of heredity lies in the nucleus. The chromatin in the nucleus contains both protein and nucleic acid, and this caused a debate over whether protein or DNA is the genetic material. Albert Kossel showed that nucleic acid is made of 4 nitrogenous bases: adenine, guanine, thymine, and cytosine. In the 20th century, the Rockefeller Institute was established in 1904. It is the first institution in the United States devoted solely to biomedical research. John D. Rockefeller's grandson died from scarlet fever in January 1901, and the capitalist and philanthropist moved to formalize plans for the research center. The labs were opened in 1906 on the site of the former Schermerhorn Farm at York Avenue and 66th Street, and I used to work there, actually. The Rockefeller Hospital which is crucial to the Institute's mission, opened in 1910. This hospital was the first center for clinical research in the United States, a place where researchers could bridge the gap between science and the bedside, and studying diseases in both the laboratory and as they manifested in patients. The Rockefeller Institute became a center of nucleic acid research. Today, it has been renamed the Rockefeller University and is one of the foremost research centers in the world, contributing to 21 Nobel Prizes as well as numerous other awards. The university's research has achieved many of the last century's seminal advances in biomedical sciences, including discovering that DNA is the basic material of heredity in 1944, determining that cancer can be caused by a virus in 1911, also confirming the connection between cholesterol and heart disease in the 1950s, and developing methadone maintenance for people addicted to heroin in the 1960s. In the 1990s, Rockefeller was crucial in developing the AIDS cocktail drug therapy, which has saved many. One of the major questions for the early 20th century in terms of how genetic information is transmitted is the heritable substance in the nucleus protein or DNA? And we're going to discuss the debate that went on in science during that period. So the tetranucleotide theory was proposed by a Russian scientist who immigrated to the US to escape persecution. In 1905, he was hired by the Rockefeller Institute to head the Department of Biochemistry. He published 700 papers, and in 1910, this scientist, Aaron Levine, proposes the tetranucleotide theory. Levine discovered that nucleic acid is made of a sugar, phosphate, and a base. Together, they form a nucleotide. He assumed a 1 to 1 to 1 to 1 ratio of A to G to C to T, and that the 4 nucleotide unit repeated over and over. Proteins are made of 20 amino acids and are therefore more complex. So he thought that protein must serve as genetic material. The transformation phenomenon that was observed by Frederick Griffith, an English physician in 1928, was something that led us away from this idea that protein is the heritable material. Frederick Griffith was interested in Streptococcus pneumoniae. He observed that virulent strains were producing polysaccharide and therefore looked smooth, and these were the S strains for smooth. Those that were mutated to a non-virulent form produced a rough-looking colonies, and these were called R for rough. He found that if he injected smooth colonies into mice, they died. And here are some of the experiments. So if you take the virulent bacteria, you inject them into mice, they die. And that's the smooth type. If you take the rough bacteria, you inject them into mice, the mouse lives. If you take the heat-killed smooth bacteria, so this is smooth bacteria that you've killed, basically. It's not alive anymore. The mouse lives. But he did another experiment. He added the rough bacteria to the heat-killed smooth bacteria, and he found that the mouse died. So how did this transformation occur? So here's another example of the experiment here on the left. Virulent kills the mouse, avirulent, the mouse lives. Heat killed, virulent, the mouse lives. Griffith's experiment shows that if you mix heat killed virulent with living non-virulent, you can get death of the mice, and you can recover living, smooth colonies from these mice. So Griffith's explanation was he did not kill all the bacteria when he boiled the smooth bacteria, but he ruled this one out by infecting mice with the heat-killed smooth bacteria alone. Perhaps the avirulent strain, the rough strain, mutated to a virulent form, but this would be a very rare event. Another idea would be that somehow the avirulent strain got transformed by the killed virulent strain. And he called this a transforming principle. But this principle, it must be heat resistant on some level. He thought it may be some substance from the polysaccharide coat. So there was some skepticism about this transformation phenomenon. And Oswald Avery came to Rockefeller in 1913 to study the differences in virulence among strains of pneumococcus. And he wanted to demonstrate what was the transforming principle. So he did this with a number of other scientists who were also at the Rockefeller, Colin MacLeod and Macklin McCarthy. And after 10 years of experimentation, doing extremely impressive and classic experiments, they published in 1944 what the transforming principle was. And I'm going to go through the experiment now that they did. They took the smooth cells that were virulent, they centrifuged them, they spun the smooth virulent cells to the bottom of the tube. They heat killed them, and they homogenized these cells. And then they treated it with either a protease, a ribonuclease, or a deoxyribonuclease. So they did several experiments. They took the rough cells, which are avirulent, and they added the smooth filtrate that they got from the first part of the experiment. And then they showed that transformation occurs. And this was a control experiment. They knew this would happen, and this is what they did on the right. This just demonstrates that the smooth cells contain the active factor that can cause a transformation to make the cells, the rough cells, virulent. Then they put the rough cells with a protease-treated version of the filtrate, and transformation does occur again. So they concluded that the active factor is not a protein because the protease destroyed all the proteins in the filtrate. Then they did another experiment where they treated the filtrate with RNase, and this destroys RNA. They were still able to transform the rough cells into the virulent smooth cells. So they concluded that the active factor is not RNA. So the final experiment was they treated the filtrate with a DNase, which destroys DNA. And they found that no transformation occurs. So from this, they concluded that nucleic acid is the fundamental unit of the transforming principle. Macklin McCarthy went on to strengthen his results. So he had used a crude DNase in the original experiment. He went on to purify and crystallize for the first time the DNase enzyme to absolutely verify that the genetic material was DNA. DNase didn't work well. This experiment doesn't really work. This helped to quiet some of the skepticism of some people that still protein was the transforming factor. He got numerous awards, including the highest scientific award in the US, the President's National Medal of Science. In 1952, Martha Chase and Alfred Day Hershey performed the definitive experiment that showed that DNA was in fact the genetic material. And here they are. Alfred Day Hershey went on to win the Nobel Prize for his insights into the nature of viruses in 1969, along with Max Delbruck and Salvador Luria. So Hershey and Chase did this very important experiment with using phage. So they knew that viruses were made of DNA and protein, and that phages attach to the host cell. Phages are also made within the bacteria before the cell lyses. DNA has phosphorus, a good amount of phosphorus, and protein also contains a lot of sulfur. So they added radioactive isotopes to the medium that E. coli was grown in. So they had one culture that had radiolabeled phosphorus, P32, and that would label DNA. And then they had one culture, which is in green here, radiolabeled with radioactive sulfur. They grew the phages into each of these cultures individually. Then they harvested the phages that came from these radiolabeled cultures. They used them to infect unlabeled bacteria. And then they tested where the labels went. So they removed the phage ghosts that don't have any DNA in them, and they checked where the radioactivity went. So they found that the ghosts were unlabeled when P32 was used, but the bacteria was labeled with P32. When they looked with S35, the ghosts were labeled. So the ghosts were basically the capsid and all the rest of the virus of the bacteriophage, but not the DNA. But the bacteria were not labeled. So it showed that DNA was the genetic information that was transferred by phages. So this was a definitive proof that DNA is capable of transferring genetic information on its own. Erwin Schargroff was an Austrian who worked at Columbia University. In 1950, he discovered that no matter what tissue from an animal he looked at, the percentage content of each of the 4 nucleotides was the same. So if you look at this table, the percentage G guanine equals the percentage C cytosine, and the percentage adenosine equals the same percentage of thymine. So the ratio of the two are the same. The percentage of G or C or A or T could vary between species. However, the ratio of G to C and A to T was always maintained. It was around 1. So this suggested that the structure of the DNA was specific and conserved in each organism, even though the sequence was not exactly the same. The next important step was to crystallize DNA. So to do this, an X-ray source passes X-rays through a lead screen. This beam of X-rays hits the crystallized protein or DNA that you're looking at. In this case, we're looking at DNA. And there's a photographic plate that records the diffraction pattern that the crystal creates when it's hit by beams of X-rays. And this diffraction pattern tells you a lot about the structure of the DNA or the protein that you're looking at. So in 1947, Ashbury started studying DNA using this method. It was a fairly common method to study the structure of proteins and other organic molecules. Rosalind Franklin then joins King's College, and Sir Bragg asked her to determine the structure of DNA. Now, Rosalind Franklin is one of the initially unsung heroes of the DNA story. So she was born in Cambridge. Her father wanted her to be a social worker, but she wanted to be a scientist. She obtained a PhD in physical chemistry, studying carbon and graphite microstructure, and spent 3 years in Paris studying X-ray diffraction techniques. She went to work for John Randall, who asked her to study DNA. Her peer at Randall's group was Maurice Wilkins. And this is, being a peer is similar to being a PI in a lab here in the US. Her analysis produced this amazing photo, which is an X-ray diffraction photo of structure B. And it's called Photo 51, and it was taken on May 2nd, 1952. And this was taken by Rosalind Franklin and R.G. Gosling. Photo 51 revealed a lot about DNA. The X that you can see here shows that this type of diffraction can come from a helical structure. The diamonds that you see here come from helix repeats, a continuation of the helix, and arise from regular series along the molecular axis. It also indicates that the sugar phosphate backbone lies outside the molecule and the bases are inside. These layer lines are helically repeating structures, and smearing that you see here is scattering from the structure parallel to the helix. It forms a symmetrical pattern that makes twists at regular intervals. So there are some missing smears in layer 4. And this is consistent with DNA being a double helix. So now, looking at all these things, what did they know at this point? They knew there were 4 bases: adenine, guanosine, thymine, and cytosine. Avery, MacLeod, and McCarty found that DNA is a transforming principle. Chase and Hershey found that DNA is the genetic material. Levine had shown that DNA is made of sugar, phosphate, and a base, and that there are 4 types of bases, A, T, G, and C, which are shown above. Chargaff showed that in each organism, the percentage of G's and C's is equal to the percentages of A's and T's. Rosalind Franklin showed that DNA has a sugar phosphate backbone, that it's double-stranded and helical. And Pauling showed that the gene might consist of 2 mutually complementing strands. Watson and Crick step in and decide to do some modeling. And in 1953, they produced a model of DNA. Watson and Crick faced competition from Linus Pauling, who was a chemist and also a crystallographer. And to create the DNA model, Watson and Crick had this key insight that A bonds with T and G bonds with C. And this led to their Nobel Prize for discovering the double helix and the structure of DNA. As the 20th century moved on, we have discussed that Levine proposed the tetranucleotide theory. Griffith demonstrates a transforming principle. Avery, MacLeod, and McCarty demonstrate that the transforming principle is DNA. Ashbury begins his X-ray diffraction studies of DNA in 1947. In 1948, Chargaff and colleagues discover the regularity in base ratios of DNA. In 1952, Hershey and Chase demonstrate that DNA is genetic material in bacteriophage. In 1952, Rosalind Franklin showed that DNA is double-stranded and helical. And Watson and Crick devised a secondary structure for DNA in 1953, based in a large part upon Franklin's work and the works of all the others that are shown here. In 1956, Frankel, Conrad, and Singer show that some viruses use RNA as genetic material, which adds sort of another dimension to this story. But in 1962, the Nobel Prize for this amazing double helix model goes to Watson and Crick, as well as Wilkins. But Rosalind Franklin was not even thanked. So, of course, this has caused a lot of controversy. Why was she ignored? Does it have to do with her gender? In fact, she didn't even live to see the Nobel Prize because she died of cancer, in part probably because of the work she was doing with X-ray crystallography, which is a little bit dangerous. But yes, here it is, the double helix. This understanding of how DNA formed the genetic basis of heredity, DNA is definitively shown to be the molecule that stores genetic information and transmits it to the next generation. So now let's take a break. You can look at this UV print by the artist Jeff Elrod. Relax, take a break, come back in a minute, and we're going to discuss a little bit more about DNA and RNA. So after all of these experiments, we now know that DNA is the genetic material. Can RNA also be genetic material? There is evidence for this from a few viruses, and I alluded to that before in the historical timeline. RNA can be put into a capsid and then released from the capsid inside the cell and then be repackaged by the cell again during duplication inside the host. Heinz, Frankel, Conrad, and B. Singer showed this could happen in the tobacco mosaic virus. And they realized that you can separate the genetic material and the protein code, the viral capsid, and remix them. So, for example, you have a type 1 tobacco mosaic virus and a related virus, let's say type 2, the Holmes ribgrass virus. These can enter a host cell. They can be packaged into each other's viral capsid, creating two types of viruses. So the progenies do not resemble the virus that donated the protein code. So let's delve a little deeper into the structure of DNA. It's typically a long molecule. It is known as a macromolecule. It's packed tightly, has three levels of complexity. Primary, the nucleotide structure and how they are joined together. Secondary, the stable three-dimensional configuration, its double helix. And tertiary, the complex packing arrangement. Nucleotides are made of three parts: the sugar, the phosphate, and the nitrogen-containing base. So ribose and deoxyribose are the sugars. And these are pentose sugars with 5 carbon atoms. Ribose is the sugar that is in RNA, and deoxyribose is in the sugar that is in DNA. The extra oxygen in ribose makes RNA a little less stable because that oxygen is more reactive. The bases are the purines, which are adenine and guanine, and the pyrimidines, cytosine, thymine, and uracil. Thymine is present in DNA, and uracil is present in RNA. So there are no T's in RNAs, there are U's in RNAs. Here's a closer look at the basic structure of the purine. And you can see that these are slightly larger molecules. Adenine and guanine are the two purines. So these do not pair with each other. They pair with another pyrimidine, which is a little bit smaller. And the pyrimidines that they can pair with are either cytosine, thymine, or uracil. So of course guanine and cytosine pair, and adenosine and thymine pair. A nucleotide, in the case of a deoxyribonucleotide, which is what makes up DNA, has three components: the base, the sugar, and the phosphate group. So we have a deoxyadenosine monophosphate on the left and a deoxyguanosine monophosphate on the right. The other nucleotides that you can make for DNA are, of course, the deoxythymidine and the deoxycytosine. To form DNA, you have to make a phosphodiester bond. And you can see here that nucleotides form a polynucleotide strand that has a direction, this polarity, this 5-3 polarity, that is given in part by the phosphodiester bond. So the 5' end has a phosphate on it, and the 3' end does not. So this is our two ends of DNA, the 5' end and the 3' end. Now, DNA also has a secondary structure because you have two strands that are in an antiparallel orientation, and that's what Watson and Crick figured out. The guanine and the cytosine pairing here, and then the adenine and the thymine pairing. And you can notice a difference in the number of hydrogen bonds. So that adenine-thymine base pair only has 2 hydrogen bonds, whereas the guanine-cytosine pair have 3 hydrogen bonds. Guanine and cytosine have a tighter association with each other. So you have the 2 hydrogen bonds here for the adenine-thymine and 3 hydrogen bonds here. The two strands run in opposite directions, so they are anti-parallel. One into the 5-3 direction, the other in one orientation, and the other one in the opposite orientation, right? But these strands are complementary to each other. You always have T on one side and A on the other, for example, or G and C. Here's a look at a strand together, the double helix, and you can see that the diameter is about 20 angstroms, and one complete turn is 34 angstroms, and there's a major and a minor groove, and a sugar phosphate backbone that stitches the nucleotides together. The base pairs, the nitrogenous base pairs, help form the helical structure as well. You can see here you got C's and G's together and A's and T's, and then the backbone is connected by all these phosphate bonds on the sugars. This creates a minor groove and a major groove. And now I'm going to show you a video of how DNA forms this helical structure.
So you're probably familiar with the general appearance of DNA as it's represented here in this computer model. Make it active. It's a double helix of 2 polymeric strands. Now, if we unwind those strands, we can see the stacked nucleotides that form this linked structure. Now, what I'm going to do is take 5 of those nucleotides out of the DNA helix in diagrammatic form and bring them out here so that we can go through them in just a little bit more detail. So, as I mentioned, DNA is a polymer of linked nucleotides. And these nucleotides are little repeating units that go down one strand, and on the other strand, they go the other way. So these are anti-parallel strands. We'll get into why that is in a minute. The major structural components of DNA is that they all share a deoxyribose sugar backbone and nitrogenous bases. The nitrogenous bases are the information carriers of the genetic material. The nucleotides themselves are linked together by phosphodiester bonds, and if we zoom in here, you can see why DNA has a negative charge. Now, these DNA molecules have these nucleotides that are linked in a directional manner. So if we zoom in on the ribose, you can see that the linkage of the phosphodiester bonds occurs at the 5' carbon, and you can see the numbering of the carbons around the ribose here, as well as the 3' carbon. And so DNA is inherently directional. We read and write DNA from the 5' direction to the 3' direction. That's not coincidentally the direction in which enzymes which replicate DNA also extend a growing chain. All right, now back to the nitrogenous bases. There are two kinds. There are purines, adenine and guanine, and there are pyrimidines, thymine and cytosine. And there are also rules governing which can bind to which by hydrogen bonding, these so-called base pairing rules. And in the base pairing rules, cytosine can bind with guanine, and adenine can bind with thymine. And this forms the basis of the genetic code. Now in RNA, thymine is actually uracil, small point. Now a beautiful little feature of knowing this hydrogen bonding pairing rules is that if you know the sequence on one strand of DNA, you automatically know the sequence on the other strand of DNA. So if this reads ACGTT cross 5' to 3', on the other strand going the other direction, it's going to read AACGT. Now put these back into the helix. I'll tuck them in. So here are the nucleotides. We'll put them back in there and twist it back up into this helical, right-handed helical spiral of antiparallel strands, where you can clearly see that the bases form sort of a semi-stacked ladder, like steps on a staircase.
So how does DNA fulfill all the characteristics of being the genetic material? It's capable of storing large amounts of complex information, the instructions to build an organism. The bases fulfill this role. This must be stable. There's hydrogen bonding between the bases. And it can replicate faithfully with high fidelity because it has a complementary strand that ensures this. So you basically have two copies of the sequence in a way. It has the capacity to vary, and this is through mutation, which we can discuss later on in the course. And DNA must encode a phenotype, a mechanism to make a product, an ability to translate the message coming from the DNA into products. And here comes the central dogma of molecular biology. So you have DNA which can replicate and be transferred on to the next generation. Genes are transcribed from DNA into RNA, and then they are translated at the ribosome into protein where they can fulfill their function. This was formalized by Crick. However, there is an alternative pathway that can occur. And this was discovered by Howard Temin, and he discovered reverse transcriptase. And for this, he won the Nobel Prize. So it is possible for RNA to undergo replication and be reverse transcribed into DNA. This happens most famously with retroviruses like HIV. So here's an example of two complementary strands of double-stranded DNA. You see the 5 to 3 orientation that both strands have, and they're an anti-parallel orientation. So the polarity of the strands is determined, in a sense, by the 5' phosphate group and the 3' OH group. The length of the DNA that is shown here is 20 base pairs. The RNA that it would encode would be this. And you can see that the A's and G's and C's are all the same, but the T's are uracil and RNA. And RNA is single-stranded under most circumstances, and this is the form it needs to be in for proper translation to occur. DNA can also assume multiple secondary structures, the most famous of which is the double helix, and it refers to its three-dimensional configuration. And these secondary structures can change with sequence and condition, and also from the regulation of histones and other things like that. Watson and Crick's model was the B DNA structure, which is a right-handed helix that forms a clockwise spiral. And you can see here the different forms, the A form, the B form, and the Z form. The A form is present when there is less water around. So the structure becomes more shorter and tilted, and it's not natural, so you don't see the A form. The Z-DNA form is a left-handed helix, and it does exist in nature. It's dictated by sequence, and this is caused by stretches of alternating G's and C's. And this has a role in gene regulation. Here's a comparison of the three different types of DNA secondary structure. And you can see that A occurs in a low percentage water situation. Although the helix direction is the same as the normal form, the B DNA structure, it is slightly more compacted, so you have more bases per turn. And the diameter is a little bit wider. But the distance between adjacent bases is shorter. Now, zDNA, which does occur in nature, has these alternating purine and pyrimidine bases, which are the G's and C's, which I discussed before. The helix direction is left-handed, so it's in the opposite type of helix. It has even more base pairs per turn than the other two structures. Rotation is in the opposite orientation. The distance between adjacent bases is larger, but the diameter is smaller. So basically, it is an elongated and narrow structure of DNA. DNA can also assume other secondary structures, and this is true of single-stranded DNA. You can have inverted repeats that cause hairpins or stem loops. And you can see two different examples where you have, in the left, an inverted repeat that has a hairpin or a stem and loop. It's also called stem loops. Or you have an example on the right where you have an inverted repeat where you just get the stem, but there's no loop. This occurs extensively in RNA, where RNAs form all these secondary structures. And this leads to the RNA's function, actually. So RNAs can simply be the message, in a sense, that causes the translation of genetic information into protein, but they can also function as biomolecules on their own. And this is caused by the secondary structures that they can form. A great example of this is the 16S ribosomal RNA, which makes this beautiful secondary structure. So here are some genetic terms and concepts that I think that you should know. Nucleotide, a repeating unit of DNA or RNA made up of a sugar, a phosphate, and a base. Purine, a type of nitrogenous base in DNA and RNA. Adenine and guanine are purines. Pyrimidines, a type of nitrogenous base in DNA and RNA. Cytosine, thymine, and uracil are pyrimidines. X-ray diffraction, a method for analyzing the three-dimensional shape and structure of chemical substances. Crystals of a substance are bombarded with X-rays, which hit the crystals, bounce off, and produce a diffraction pattern on a detector. The pattern of the spots produced on the detector provides information about the molecular structure, and this was used to understand the molecular nature of DNA. Antiparallel refers to a characteristic of the DNA double helix in which two polynucleotide strands run in opposite directions. Transcription, process by which RNA is synthesized from a DNA template. Translation, process by which a protein is assembled from information contained in messenger RNA. The central dogma, concept that genetic information passes from DNA to RNA to protein in a one-way information pathway. Replication, process by which DNA is synthesized from a single-stranded nucleotide template. Next time we will discuss DNA replication. I hope you've enjoyed the lecture, and I will talk to you soon.